Gopinath Balaji, and I'll be presenting our research, which is detection and classification of brain tumors using deep convolutional neural networks. I'll begin by stating our objectives and give a brief introduction about our topic. Then we'll move on to our proposed methodology, which covers our architecture diagram, reprocessing techniques, and our model specification. Finally, I'll conclude by stating the results that we have obtained and some possible improvements that can be made to a model in the future. Our main objective for this research was to apply various image pre-processing techniques to improve the quality of the input data for the model. Our next aim was to implement various famous CNN architectures using transfer learning so that we can differentiate and classify between normal and abnormal tissues with high accuracy. Since our research mainly revolves around brain tumors, we need to first understand what exactly a tumor is and what are the types of tumors and how are tumors detected. Normally, cells grow and divide to produce new cells in a controlled and orderly manner in a body. But a tumor is formed due to swelling or morbid enlargement that results from an overabundance of cell growth and division. All tumors are classified into two main types, benign and malignant. A benign tumor is a non-cancerous tumor. That means it does not invade nearby healthy tissues or spread throughout the body. However, malignant tumors are cancerous. They have cells that are abnormal and divide uncontrollably without order, so they often spread to other parts of the body and damage nearby tissues. Now coming to brain tumors, there are three main types: meningioma, glioma, and pituitary tumors. There are various imaging methods to identify these tumors in the body. The most common one used by medical professionals is magnetic resonance imaging. It is a non-invasive method for doctors to produce high-resolution images of the inside of the body that helps diagnose variety of problems. Now we'll move on to proposed methodology, which covers our architecture diagram, pre-processing techniques, and our model specifications. The data set we have used for our model is an open source data set available on Kaggle. It consists of approximately 3,000 MRI images and 4 classes. 3 of the classes are meningioma, glioma, and pituitary tumors. The 4th class are the images with no tumor. On the data set, we applied pre-processing techniques like bias correction, image denoising, and skull stripping. After the data is ready, we implemented our CNN models and evaluated them using various metrics like accuracy, precision, recall, and F1 score. Our first pre-processing method is bias correction. Bias field signal is a low frequency and very smooth signal that corrupts an MRI image. This is especially the case for images generated by old MRI machines. To correct the bias in an MRI scan, we have used N4 bias field correction using the simple ITK Python package. N4 is a short form of non-parametric, non-uniform intensity normalization. The N4 bias field correction algorithm is a popular method for correcting low frequency intensity and non-uniformity present in the image. In any bias correction process, the effect is often too subtle to observe visually, but it becomes more apparent by flipping back and forth between two images. This is even true for more sophisticated software like bias field correction offered by BrainSuite. Our next pre-processing method is image denoising. Image denoising refers to the process of removing noise from a noisy image in order to recover the original image. However, because of some components like noise, edges, and texture, it becomes difficult to differentiate them throughout the denoising process, and denoised pictures may unavoidably lose some features. We have tested 3 different denoising methods. The first one is Gaussian filter denoising. A Gaussian filter is a linear low-pass filter used to reduce noise and blurring region of an image. Here we see that the effects of denoising an MRI image using Gaussian function. To the left, we have the original image, and towards the right, we have an image after, we have the noisy image to the left, and we have a denoised image towards the, so this is basically what it is. Total variation denoising is a noise removal process which is based on the principle that signals with excessive and possibly spurious detail that have total variation. According to this principle, reducing the total variation of signals while preserving important details such as edges. So these three types of denoising techniques, BM3D performed the best. Here is an image of total variation denoising. Yeah, now we finally, after removing the bias and noise, skull stripping is performed. Skull stripping is an important step to pre-process the images. It removes the unwanted part of the MRI image like extra tissues, cortical surface, etc. We apply Ochu's method of image thresholding, which will give us a binary mask of the image. Using the threshold, the brain can be extracted by finding the largest connected component. But sometimes when the image is noisy, the extracted brain image might contain many holes in it, which is why we also use a closing transformation to close the holes and extract the brain properly. The first image shows the input image. The second image shows an attempt to extract the brain without applying closing transformation. And the last image shows the final image after applying closing transformation. As visible from the final image, skull stripping has allowed us to reduce noise considerably. Before we get to the results, here are a few hyperparameters that we have set for the model. Since our dataset contains only about 3000 images, we have used data augmentation to increase the size of the dataset. Instead of adding multiple dense layers towards the top of the model, we have used global average pooling and dropout of 0.2 to reduce overfitting. The initial learning rate is very low initially because a high learning rate may overshoot and not converge. To improve the accuracy of the model, we have also used reduce LR on plateau, which is a function offered by Keras. It reduces the learning rate when there is a drop in metric that we are measuring. Coming to the results, we tested our methodology with 5 different models, which are ResNet 50, Exception, VGG16, MobileNet V2, and EfficientNet B0. As the table says, our best performing model was EfficientNet B0. The accuracy it gave was 97.61%. Our worst performing model was VGG16. The reason it did not perform well is because it lacks skip connections that reduces information loss as we go further deep down the model. This is the graph we have obtained for our best performing model, EfficientNet B0. To the left, we have the training and validation accuracy graph, and to the right, we have the training and validation loss graph. This table shows the metric we achieved for each class in the data set. We achieved 100% accuracy for pituitary and no tumor classes, and we achieved around 99 to 97% accuracy for meningioma and glioma tumors respectively. Here are some papers that we have used for reference. To conclude, by using various pre-processing techniques and methods to prevent overfitting, we were able to achieve a high accuracy of 97%. But there are various improvements possible. We have only used CNN to classify brain tumors, but various detection algorithms like YOLO exist that will show the actual location of the tumors with bounding boxes. This will further make it easier for medical professionals to diagnose the patient. Another improvement that is possible is that traditional machine learning classifiers like SVM can be used instead of using dense layers followed by softmax. Thank you for listening.
Thank you very much, sir, for your presentation.
Thank you, sir. Yeah.
Thank you.